So today we are going to talk about the structure of root apex. What is a root apex? A group of initials present at the subterminal region of the growing root is called root apex. Okay, so basically root apex, these are the group of initials and they are subterminal in position because of presence of root cap. Now what are the characters of root apex? What is its structure? It is embryonic in origin. Means the meristematic cells, this zone that is formed within embryo, it's responsible for growth of primary root. The initial growth of the root, which is called primary root, that is because of root apex. It is inverted cup-like structure it is shorter than shoot apex and also lack lateral appendages like leaves, branches, etc., as we see in case of shoot apex. It is protected by root cap at the tip. Cell division in meristem or meristematic cell adds new cells in both directions to form body of root and root cap. There is no distinction between node and internode in the root. Thus, it shows uniform growth in length. The cells have dense protoplasm and prominent nuclei. Cells are in the state of active division. So these are the characters of root apex. First thing, it is embryonic in origin. It is responsible for the growth of primary root. Its inverted cup-like structure, shorter than shoot apex. It lacks lateral appendages. It is protected by root cap at the tip. Then it is going to add new cells in both the directions, that is towards upper side it will contribute to body of root and towards lower side to root cap. Then the root apex has no differentiation of nodes or internodes, so it's cylindrical in shape. The cells have dense protoplasm and prominent nuclei, and they are in the state of active division. These two, they are the typical characters of a meristem. So that was related to the general structure. Now we are going to draw the cross-sectional view of root apex. So initially you have to make a cylindrical area. Draw the dotted lines because we are going to form cells in it, and then you will taper it to form a dome-like tip. Okay. Then we are going to specify the central area. In the central area, just from this point on both the sides, we will converge the lines towards center. Then you will make these lines double, and as move towards center, slightly narrower them this way. Similarly along this side also, initially the line should be broader, and as we turn it, the line becomes narrower. So this will be the main area of differentiation, and from the center almost divide the total cross section 1/3 portion as a central cylinder. Okay, and you will curve it to meet at this open end. Fine. So now we are going to draw cells in this. So initially we are starting with dermatogen. The cells are rectangular, broader, and without any kind of spaces. So between these two lines we will make the rectangular cells. As we move towards the center, they will become slightly squarish, and we will meet in the center where you have to draw two initials. Similarly on this side also, I am going to draw few cells only so that you get an idea what kind of shape we have to draw here. Okay, then you will darken this boundary, because on dermatogen we have a thin layer of mucilage, and we will converge it towards the center. Similarly along this side also, the mucilaginous sheath on dermatogen. Now the central cylinder, that is a mixture of large and small elongated cells, broad and narrow cells. So the first tier I am going to draw is of broad cells. Then as you move towards the center, you have to make the size smaller. Then another tier of elongated cells. Again I am saying no intercellular spaces should be there. Another also you can draw elongated. This is your choice, okay? You can have the alternate arrangement of elongated and broad cells. So now the next tier is broad. Next tier of elongated cells. One more elongated cells. In the center, you have to keep this thing in mind that towards these initials, the cells, they are more or less polygonal. Another row of elongated cells, broader cells. In order to differentiate this area, I am just going to darken this outer boundary. Okay, this is a central portion which is called plerome. It is going to give rise to vascular cylinder. Now the in-between area. The cells are polygonal without intercellular spaces. While drawing polygonal cells, you should not have corner of a cell into the other. Okay, it's important. Like when you are drawing the cells, you can show 4, 5, 6, 7 sides, but the corner of a cell should not be there in the other cell. Like this is fine, this is a fine arrangement. But if I draw a cell like this, so the corner of the cell lies in this cell, so then it will become a wrong arrangement. So you have to draw the polygonal cells and always use corners to draw next cells. For mixing, you can have few 4-sided cells, 5 hexagonal. Okay, there may be size variation. Same kind of cells you will draw on other side as well. And in order to differentiate these cells, we will make them dotted so as to separate dermatogen and central cylinder from this. Similarly, you will draw cells on the other side. Now we are going to draw root cap. Now before root cap, you will divide this area, make it slightly parallel and make horizontal cells without intercellular spaces. This is a region of calyptrogen from which root cap will be formed. So these are calyptrogen cells. Now afterwards you will draw circular or oval shaped cells with intercellular spaces, variable sizes. The cells should not overlap. You can slightly D-shape them to fit in the area. So it should be a dome shaped area. Okay, they should not overlap. Whenever you are drawing cells, you can slightly D-shape them in order to fit them properly. Okay, but don't draw it like this. This is wrong. There should be no overlapping. So that was about the structure. Now coming to the labeling part, this outermost layer is mucilage. Then we have dermatogen. Next layer is peribulum. Central one is plerome. Then here we have few layers of calyptrogen, and at the tip there is root cap. Okay, so this is a structure of LS of root apex. It's very easy, just you need to remember the shape of the cells, that in the central cylinder we have mixed broad and narrow cells. In between we have polygonal cells, and the outer one they are perfect rectangular cells which are broader as compared to longer. And the cells of the root cap, they are simple parenchymatous, oval or circular cells. They have intercellular spaces, but otherwise all other cells, they all different layers which are called, we'll define them later on, these are called histogens. They are without intercellular spaces. Now we mentioned here it's an inverted cup-like structure. So if you look at the overall initials in this area, in this much area we have the meristematic cells, and they look like the inverted cup-like structure. Okay, so that was all about the structure of root apex. Thank you.